Hello again. We will continue to learn about Mendelian genetics. Monohybrid and dihybrid. Gregor Mendel is a monk who plants and bred pea plants, or the scientific name is Pisum sativum, and study about inheritance of the pea plants. The reasons he chooses peas for the experiment are: first, they are easy to grow. Second, they had short life cycle. Third, their pollination can be controlled. And the fourth reason is, they have easily observable characteristics. Mendel had observed and studied about these 7 characteristics of pea plants. Each characteristic has 2 contrast traits. The seed shape has round dominant trait and wrinkled recessive trait. The seed color has yellow dominant trait and green recessive trait. The flower color has purple dominant trait and white recessive trait. The pod shape has inflated dominant trait and constricted recessive trait. The flower position is at axial dominant trait and terminal recessive trait. And last one is stem length, where tall is dominant trait and dwarf is recessive trait. Mendel had conducted monohybrid cross and dihybrid cross. Monohybrid cross is a crossing or mating or breeding that involves one character which is determined by one gene. For example, a cross involved flowers color only, and a cross involved peas height only. Meanwhile, dihybrid cross is a crossing that involves 2 characters which are determined by 2 genes, that located at different chromosomes. For example, a cross involved seeds color and seeds shape, and a cross that involved peas height and flowers color. For each monohybrid and dihybrid cross, Mendel's had conducted self-cross and test cross. In monohybrid self-cross among F1 or first generation, Mendel's found out that the phenotypic ratio of F2 or second generation is 3:1. From the founds, Mendel had proposed his first law, which is law of segregation. In monohybrid test cross, Mendel found out that the phenotypic ratio is always 1:1, if the dominant phenotype is heterozygous. In dihybrid self-cross among first generation, Mendel found out that the phenotypic ratio is always 9:3 to 3:1. From the founds, Mendel had proposed his second law, which is law of independent assortment. In dihybrid test cross, Mendel found out that the phenotypic ratio is always 1:1 to 1:1, if the dominant phenotype is heterozygous for both genes. Let's look at the monohybrid self-cross. Firstly, Mendel made a crossing between 2 true breeding individuals as parental generation. True breeding is homozygous or having identical alleles. So, true breeding purple flower was crossed with true breeding white flower. Each parent individual produce gamete by meiosis. Please take note, you only have to show the type of gamete. For example, true breeding purple can produce one type of gamete only, which carry dominant allele. True breeding white can produce one type of gamete only, which carry recessive allele. When the 2 gametes from each parent undergo fertilization, first generation will be produced, where all first generation are heterozygous purple. Then, the first generations were self-crossed, where heterozygous purple crossed with heterozygous purple. Each heterozygous purple produced 2 types of gametes, one carries dominant allele and the other one carries recessive allele. When gametes from different parents undergo fertilization, second generation will be produced. The second generations produced are homozygous dominant purple, heterozygous purple, and homozygous recessive white. The genotypic ratio of the second generation is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Meanwhile, the phenotypic ratio is 3 purple to 1 white. The second generation shows there are 3 out of 4 are purple and 1 out of 4 are white. This shows that first generations carry a recessive allele that control white color and pass it to the second generation, therefore white flower presence in second generation. In order to pass the alleles in first generation must separate or segregate into single gamete. During Mendel's time, he used factor term, and later, allele was introduced. From this founds, Mendel proposed the first law, which is law of segregation. Law of segregation states that the two alleles of a gene segregate or separate from each other during gamete formation and end up in different gametes. This is a gene that consists of a pair of allele. The two alleles will segregate into different allele during meiosis. Each gamete will carry one allele. This is known as law of segregation. Let's look at monohybrid test cross. Test cross is a cross between an individual of dominant phenotype, but unknown genotype with a homozygous recessive individual. If purple is a dominant phenotype, there are two possibilities of genotype which are homozygous dominant, or heterozygous. If white is a recessive phenotype, the genotype is always homozygous recessive. Which means, we know the genotype for recessive phenotype, but we don't know the exact genotype for dominant phenotype. Therefore, test cross is used to test whether the dominant phenotype is homozygous or heterozygous genotype. Let's make a test cross between homozygous dominant purple with homozygous recessive white. This crossing produce one type of offspring or progenies only, where all are heterozygous purple offspring. It means that, if all the offspring or progenies are purple, the genotype of the dominant phenotype is homozygous dominant. Now, let's make a test cross between heterozygous purple and homozygous recessive white. This crossing will produce two types of offspring, which are heterozygous purple and homozygous recessive white. The genotypic ratio of the first generation is 1 heterozygous to 1 homozygous recessive. Meanwhile, the phenotypic ratio of first generation are 1 purple to 1 white. It means that, if half of the offspring or progenies are purple and half are white, the genotype of the dominant phenotype is heterozygous. This is similar to Mendel's expected phenotypic ratio for monohybrid test cross, which is 1 to 1, if the dominant phenotype is heterozygous.